Under the title of Molecule of Life, we are going to make an experiment about a few main molecule of life, which is carbohydrate, protein and lipids. The objective of the experiment is to identify or test what type of carbohydrate, amino acid and lipids.
These are the materials and equipment that we use while conduct this experiment.
Firstly, carbohydrate of Molich test.
5 drop alpha naphtha solution, 5% were added to the dose tubes. And 1 ml concentrated sulfur acid were added into each tube. And make sure to not shake them.
Buffer test. 5 ml buffer tester was placed into 4 different test tubes. 2 ml of sucrose 2%, glucose 2%, maltose 2% and starch 5% was added into each test tube separately. All of them were soaked in boiling water for 30 seconds. The result was recorded.
Benedict test, which is reducing sugar. Firstly, 1 ml of sucrose 2%, glucose 2%, maltose 2% and starch 5% were added onto each test tube separately. The test tube was specifically labeled. Next, 2 ml Benedict solution was put into each test tubes and mixed them by gently shook. Furthermore, heated briefly in boiled water for 2 minutes. The result was observed. For polysaccharide, firstly, 5 drops of sucrose 2%, glucose 2%, maltose 2% and starch 5% was placed onto tie patches. And then 1 to 2 drops of iodine potassium iodide solution was added.
Burette test. Firstly, 3 ml albumin solution 5% and milk were placed onto 3 different test tubes. 3 ml sodium hydroxide 2% was added and mixed gently. 4 drop cuprum sulfate 0.5% was added and observed the color change. Melon test. Firstly, 3 ml albumin solution 5% and milk were added onto 3 different test tubes. Added 3 drops of melon solution. Lastly, observed and recorded the result.
Acrolein test. 5 drops of coconut oil, palm oil and vegetable oil was placed into 3 different test tubes. Next, 1 gram of sodium bisulfate was added and heat slowly using Bunsen burner.
The smell present was indicated and the result was recorded.
Coloring test. First, 3 drops of coconut oil, palm oil and vegetable oil was placed into 3 different test tubes. 5 ml of distilled water was added and mixed by gently shaking. Lastly, a few drops of Sudan ivy was added. Carefully mixed by hard shaking. Left them for 1 minute and the result was observed.